Hi, good morning. By this presentation, I wanted to give the details of the general characters of enzymes for you. By the end of my lecture, you should be able to define the enzymes and other related terms like active site, apoenzyme, coenzyme, cofactors, prosthetic group and enzyme specificity etc. So, these are the terms you might have known earlier in your classes, but we will discuss again in detail. And you are also able to describe the characters of the enzyme at the end of my lecture. So, coming to the first of all, I wanted to make you know what are the enzymes. These enzymes are present everywhere in living organisms. Daily we are taking food and this food is in the form of a carbohydrates, lipids and proteins. There is a system exclusively present in living organisms, that means animals, called as a digestive system. But taking of the food by the digestive system is unable to finish off this process, that is called digestion. So, there are some substances by which the digestion process will be completed. For example, carbohydrates will be digested by a substance called amylases, etc., sucrases, like that, maltases. Several enzymes are present for the digestion of the carbohydrates. Similarly, proteases digest proteins, lipases digest the lipids. Like several enzymes aids in the process of the digestion. Take the process like photosynthesis, respiration in plants. We know that photosynthesis completed by two reactions, that is called as the light reaction and dark reaction. Light reaction will be completed by the light, which is the physical phenomenon, whereas the dark reaction is completed by the biochemical reactions. So, in this biochemical reaction also certain substances plays an important role in the completion of the processes. So, in all these reactions, that means in all the biochemical reactions, these substances helps in the completion of the processes. That is why enzymes are called as the biological molecules acting as a catalyst, which are proteinaceous in nature. So, they catalyze all the chemical reaction without altering its structure at the end of the reaction. They also help increase in the rate of the reaction by lowering the energy of the activation. So, these are the molecules acting as a catalyst. Then there are also some other term, that is catalyst. We called enzymes as a catalyst, biocatalyst, but there are some other catalysts which are called as the chemical catalyst, helps in the finishing of the chemical reactions. So, let us examine the differences between enzyme catalyst and chemical catalyst. So, these coming to the enzyme catalyst, I already told they are proteinaceous in nature, whereas chemical catalysts are non-proteinaceous in nature. These biochemical catalysts are specific reaction, whereas chemical catalysts have different reactions. So, they occur, enzyme catalysts occur via active sites of the enzyme. They does not return to the original state after a biochemical reaction. Generally, the biochemical enzyme catalysts are produced by living cells and acts inside only in living cells, not elsewhere, whereas the chemical catalyst always returns to its original state and reacts outside living cells. So, these are the main differences between the enzyme catalysts and chemical catalysts. So, enzyme catalysts exist only in the living cells, whereas the chemical catalysts are existing outside the living cells. So, what is the importance of these enzymes? So, enzymes are essential in chemical reactions. Without the presence of these enzymes, the chemical reactions will not be finished off. That is why they play an important role in various metabolic processes like photosynthesis, respiration, nitrogen metabolism, lipid metabolism, etc. And they are also important in the diagnosis and therapeutics in a medicine field. All the biochemical reactions are enzyme catalyzed reactions in living organisms. That's why these enzymes are very important in all living organisms. Levels of the enzymes in the blood also very important to know, because it is used as a diagnostic tool nowadays. Because in the diagnosis of the disease like myocardial infarction, the enzymes are estimated in the blood. That's why the enzymes are very important to know for biologists, and these are therapeutically used, such as even digestive enzymes. Nowadays people are taking in the form of a amylases, sucrases, lipases and proteases. So in the form of these enzymes also, the digestive enzymes are used in the therapeutic uses. So there are several important other factors for the enzymes, but I am giving only a few for the convenience purpose. So coming to the structure of the enzymes, already I told that enzymes are made of the proteins. They are proteinaceous in nature. That means they are synthesized from the amino acids. So their structure is similar to the structure of the proteins, which is a three-dimensional structure. Whereas nowadays the RNA molecules are also found which exhibits the enzymatic properties. Hence they are also called as the ribozymes. So they are very specific and they only work with a certain set of substances. So they must be fit into the active site of the enzymes, otherwise the enzymes will be inactive and they will be inefficient and the reaction will not be finished off. So they must have a suitable substrate. Unless the suitable substrate, the enzyme will not work. So this is the structure of the enzyme. That means it is made up of a proteins. So there are coming to the types of the enzyme. Based on the structure of the enzyme, there are different types of the enzymes existing in all living organisms. Say simple protein enzymes, and the other is a complex protein enzyme. Simple protein enzymes made up of simple proteins. The example is urease and our amylase, which aids in the digestive system. It is a simple protein enzyme. Whereas the complex protein enzymes, it is made up of a conjugated proteins. That means along with the protein, some other part is present, which is called as a non-proteinaceous part. The enzyme without the non-protein moiety is termed as a apoenzyme. So the complex protein has two parts, that is a protein part and a non-protein part. The protein part is called as the apoenzyme. That means without a non-protein moiety. When the apoenzyme combines with the that is non-protein part, then it is called as a holoenzyme, which will be the active enzyme. So with its non-protein component, it acts as a, along with its non-protein component, the enzyme acts on the substance. That is why holoenzyme is a active substance. Apoenzyme is the protein part, holoenzyme is protein and non-proteinaceous part. So this non-proteinaceous part, otherwise called as the prosthetic group. This prosthetic group is, it may be loosely bound or tightly bound to the protein part. So this non-protein part tightly bound to the apoenzyme, and it is present along with the enzyme in vicinity of the active sites of the enzyme, and these are bind with the enzyme by a covalent bond. So these are non-protein parts which is tightly bound or loosely bound to the protein part is called as the prosthetic group. This may be in the form of an organic molecule or inorganic molecule. The non-protein part which is in the form of a organic molecules is called as a coenzyme. For example, vitamins and compounds derived from the vitamins are called as the coenzymes. For example, coenzyme A. This is the coenzyme A which plays an important role in along with the enzyme, which is essential for the enzyme for taking part in the completion of the biochemical reactions, and the other one is cofactor, which is in the form of a inorganic form. So this non-protein chemical bond, which is in the form of a inorganic molecule, bound either tightly or loosely to an enzyme. It is also required for the catalysis. For example, hexokinase. Hexokinase requires magnesium ions. So these without this magnesium ions, hexokinase will be inactive. So this magnesium ions will be the cofactor for the hexokinase. So there are several molecules like zinc, iron, copper, cytochrome, oxidases, like that. There are inorganic substances are binding with the enzyme required for the completion of the biochemical reactions. And these are the examples, cofactors, that is for carbonic anhydrase, zinc is the cofactor, cytochrome oxidase copper is the cofactor, and this is the prosthetic group. Usually the prosthetic groups are in the form of a heme group or flavins or biotins, and the coenzymes are strongly bound and they are low molecular weight non-proteinaceous forms. The example is TPP, that is a thiamine pyrophosphate, flavin mononucleotide, flavin adenine nucleotide FAD, NAD is nicotine adenine dinucleotide, and NADP, nicotine adenine dinucleotide phosphate. So these are all the non-proteinaceous moieties binded along with the protein part of the enzyme. So these are the examples as a cofactors and enzymes. Coming to the general characters of the enzymes. So these enzymes already told they are proteinaceous in nature and helps in the biochemical reactions. But there are unique and special properties enzymes for the enzymes. So because of these properties only, the enzymes are performing several functions, unique functions. So these are the few things to mention. They are colloidal in nature, and they are catalytic in nature, and they are highly specific in performing the chemical reactions, and they need coenzymes for activity, and they work best at the optimum temperature and pH. Some catalyzes the reversible reactions. These are the general characters of the enzymes to mention. Coming to the first character, that is colloidal in nature. So usually the enzymes are of giant in size, ranging from 12,000 to over 1 million in size, that is Daltons in size. Because of the larger size, they look very smaller when compared to the substrate and active site. That is why they are very larger when compared to the active sites and substrates, and they are having very low rates of the diffusion. That is why they do not diffuse very rapidly into the water. That is why they form the colloidal substances in the water, and they are non-dilizable. But some of the enzymes are dilizable and they will be dissociated into the water in the form of a coenzymes. So how these enzymes are becoming colloidal means, because of the low rates of the diffusion, the enzymes are becoming colloidal substances in the water. So because of this giant size and low rates of diffusion, the enzymes are exhibiting the colloidal nature of the enzyme, property of the enzyme. Coming to the catalytic nature of the enzymes, all the enzymes have a unique property. That means there is a virtual absence of the side chains, and this catalytic efficiency of the enzyme is explained based on the turnover number or molecular activity of the enzymes. This turnover number is explained, that is number of the substrate molecules converted into the number of the products in a unit time by the completely saturated substrate. That means after utilization of the enzyme and saturation of the enzyme completely, the number of the saturated molecules converted into product molecules can be called as a turnover number. This turnover number differs from one enzyme to other enzymes. Here is the table for you, that is carbonic anhydrase, which is having a 6 lakhs turnover number. For example, this catalase, take the catalase example, where the turnover number is a 50 lakhs. That is a single molecule of catalase that is performs the biochemical reaction in which the hydrogen peroxide molecules converts into the oxygen and water in a minute. So, 50,000 molecules of H2O2 can be converted into water and oxygen in a minute. That is called as a turnover number, and also the molecular activity or catalytic efficiency of the enzymes. So, you can go through the table for different enzymes. We have given the turnover number and the enzyme specificity. So, these enzymes have varying degrees of the specificity because of the specific nature of the action. So, this is the property which is unique for enzymes. So, they must act only certain substances. So, they recognize and they catalyze a single substrate only. So, it cannot catalyze the other substrate. For example, sucrase acts only on sucrose, maltase acts on only maltose. So, sucrase do not act on maltose. So, maltase do not act on sucrase. So, there are other that means the enzyme also acts on a group of compounds belonging to the similar substrate. So, they do not only act on a single substrate, they can act also on a group of similar substrate. For example, on the phosphate group, on the sulfate group, on the hydroxyl groups, like that they act on group of the similar substrates. They also act as a particular type of the bond. That means optical isomers and geometrical isomers. So, based on the enzyme specificity, the enzymes are recognized as 4 types. That is absolute specificity, that means they catalyze only a single substrate. For example, urease catalyzes only hydrolysis of urea. And coming to the group specificity, they catalyze a group of compounds for similar substrates. For example, hexokinase adds a phosphate group to hexoses like glucose 6 phosphate, fructose 6 phosphate. Fructose 6 phosphate and glucose 6 phosphate are the similar substances called hexoses, but hexokinase adds the phosphate group to either glucose 6 phosphate or fructose 6 phosphate, and the other one is linkase, and they catalyze on the one of the 2 optical isomers or one of the 2 geometrical isomers of the compounds. For example, take the chymotrypsin, it catalyzes the hydrolysis of the peptide bond. So, these are in the form of catalyzation on which it is acting. That means the enzyme is acting whether on the single substrate or on the group or on the linkage of the bond. These are the enzyme specificity existing in living organisms. And this chemical that is biochemical catalysts also speed up the chemical reactions. So, this may be owing to the property of the 3-dimensional structure of the enzymes. So, the enzyme is having a 3-dimensional structure. So, not surprisingly, active site is also having the 3-dimensional structure. So, this active site is made up of different amino acids. These amino acids aids in the binding of the substrate to the enzyme. There are some other sites, that means other means allosteric site, another site. It is called as another site, allosteric site. So, the enzyme possesses 2 sites called as the active site and allosteric site. So, active site is the catalyzing site and it binds the substrate to the enzyme, and allosteric site means it is some other site where the other molecules which inhibits the activities of the enzymes can bind. So, coming to the next property, that is Thermo Lability. So, as these enzymes are made up of proteins, they are very sensitive to temperature. So, with the decrease of the temperature, that means if the enzymes are stored at the 0 degree centigrade or below degree centigrade, they become inactive. But this is reversible. That means with increase of the temperature from 0 to or from higher to 0, the activity of the enzymes is increasing, but only at certain level it increases. After reaching to a certain level, the enzymatic activity decreases. For every rise of the 10 degree centigrade, the enzyme activity rises in 2 to 3 times, which is called as a temperature quotient. That is why we can say that a temperature quotient for enzymes is 2 to 3. Q10 is 2 to 3 for enzymes. In the activity of the enzymes, because of the denaturation, that means there is a breakage of the hydrogen bond and disruption of the hydrogen bonds is there. That is why they become weak and they become inactive at a higher temperature. The optimum temperature is 35 degrees centigrade. 0 degrees it becomes inactive, where we can store the enzymes. 10 degrees centigrade for weeks, but above 30 degree centigrade it is hours, above 50 degree centigrade it is inactive. Coming to the pH sensitivity property, pH also affects the rate of the enzyme substrate complexes. Most of the enzymes are optimum pH around 7, that is isoelectric point of the enzymes. When that is acid, when we add the acids to the solutions or in the acidic medium, they do not exhibit properly because they changes their shape. Coming to the reversibility of a reaction, the digestive enzymes are having this property, that is reversibility of a reaction. They catalyze hydrolytic reactions. For example, lipase, it catalyzes and hydrolyzes. That means synthesis of fat from the glycerol and fatty acid, and this glycerol and fatty acid is synthesized into fat and catalyzed by the enzyme lipase. At the same time, the fats are dissociated into glycerol and fatty acid by the lipase. So, this is called as a reversibility of a reactions.